My name is Lara Vimercati, and I'm a PhD student in the Schmidt lab at CU Boulder. And our lab studies microbial diversities and communities at extreme high elevation. And today I will talk to you about how some of these environments, in particular the Atacama volcanoes, can represent some of the best Martian analogues here on Earth, and tell you about what we have found out of the life existing there through different approaches that go from microbial diversity community analysis to genomics. So terrestrial analogs of the Martian surface offer insights on its habitability potential. In the last decade, extreme environments that were thought to be devoid of life have actually been shown to be populated by various levels of biodiversity, supporting the notion that pretty much, at least on Earth, life always finds a way. However, we know that no analog is a perfect representation of the Martian surface, but we argue that high elevation cold desert soils are some of the best model systems for life on Mars surface. So this type of high elevation cold deserts occur worldwide on mountain ranges between the upper boundary of plant distribution and the lower limit of permanently ice-covered regions. So that's represented in this picture, where we have a relationship between latitude and elevation, and they would occur in this brown belt worldwide, and some of the highest peaks are also shown as a reference. So among these environments, the most extreme probably occur in the dry valleys and slope of the Andean volcanoes above 5,000 meters of elevation. So conditions that life has to face there is even more extreme than what we would find in the Atacama Desert. So microbial life has to cope with a complex interplay of parameters that go from high UV flux, extreme diurnal freeze-thaw cycles, low atmospheric pressure and pH in the soils, and an extremely low nutrient content and water activity. So the highest of these volcanoes is Volcan Yuyayako, which is also the highest archaeological site on Earth. So close to its summit, some of the best naturally preserved mummies have been found, and they're really interesting on a microbiology perspective because they pretty much show no sign of decay after being buried there for more than 500 years without preserving chemicals. So this inspired our first expeditions to the site to address questions about the cold-dry limit to life on Earth. So a first analysis of the soils have shown that there's nearly undetectable levels of water, organic carbon, microbial biomass, and the extracellular enzyme activities. And it seems like only a very limited spectrum of bacterial and fungal lineages have been able to overcome the harshness of this environment and possibly functioning in situ. So when we look at the distribution of the main phyla that we have in this environment, we see that they're completely dominated by chloroflexi up here and actinobacteria. So we are also below the dry limit of photoautotrophy, since no phototrophs have ever been found at these elevations. And they're probably the soils that have the lowest microbial diversity ever reported in a terrestrial system, especially if we compare it to the dry valleys of Antarctica and Himalaya. There are, however, some niches that are able to mitigate some of these environmental challenges. For example, fumaroles on volcanoes are able to provide water and warmth and CO2 through degassing, and we have seen that they support some way more complex communities, especially autotrophs. Like they form some very peculiar mat structures really close to the summit, and it is evident from the rarefaction curves of our eukaryotic communities that fumaroles support a much more a diverse community compared to the dry soils that are just surrounding them. Another oasis is represented by the so-called Nieves penitentes. So these are ice structures that are pretty common along the high elevations of the Atacamas. And they periodically release melt water around the soil. So they're pretty much the only semi-permanent sources of water in this environment. And again, we've been able to see that they are able to support much richer communities, and especially important is that we found phototrophs in the soils and the ice structures themselves compared to the dry soils where you would never find any phototrophs. So the phototrophs found are mainly the form of algae, and interestingly, no cyanobacteria have been retrieved from here. So it seems that penitentes and fumaroles are the only structures that allow the presence of phototrophs in this type of environment. Another very harsh trait that has been completely understudied, but it's very common in this high elevation environment, is the extreme diurnal freeze-thaw cycle that occurs on these soils. So as we can see, temperatures can fluctuate starting from a -10 up to 30 degrees within a 24-hour cycle. So daily temperature cycling across the freezing point is a key challenge for microbial growth and survival, as it is known to cause multiple damages on multiple levels, as it combines the stress of the cold with the reduction in water activity. So in this type of dry and cryophilic environments, it was assumed that the low biomass and diversity was mainly due to the lack of water. However, recent studies have challenged this by showing, for example, that nutrient additions in some periglacial soils in Peru and the Antarctic dry valleys have a much stronger effect than water additions on the community structures. We know that that soils in Yuyayaco have some of the lowest nutrients available. So it is entirely possible at this point that microbial life happens in the soils only if when there is water availability, also nutrients are available. So given these premises, I wondered how would these extremophilic communities respond if we partially alleviated the water and nutrient limitations in these environments under the field thermal fluctuations. So we assume that the communities lay dormant for most of the time and only come to life pretty much when there are pulses of water and the nutrients through snowfall events, which are pretty infrequent in this environment, and aeolian depositions of organic matter from the atmosphere. So the potential for these communities under occasional favorable conditions remains completely unknown to date. So what I decided to do was to apply experimental water and nutrient amendments in discrete events to soil microsystems, microcosms, and subject them to the freeze-thaw cycles that we see in the field. So to do that, I placed some soils that we collected above 6,000 meters on Yuyayaco in a temperature control chamber that is able to mimic that fluctuation in temperature that we see also in the field. And we had the two types of treatments. So we had microcosms that were exposed to water additions to simulate the snowfall events, and then freeze-thaw plus water and nutrients to simulate also occasional aeolian depositions in the soils. I also parsed out the cumulative effect of multiple watering events by simulating 3 discrete snowfall events. We also allowed the soils to dry up to 90% before applying a new water addition so that they would simulate pretty much the moisture conditions that are normally found in this environment. So after extracting our DNA, doing the sequencing of the main 16S and 18S of the community, analyzing them, what we saw is that the community was significantly changing. So in here we have a PCOA plot of the bacterial community, and we see how the community significantly changes when treatments are applied, both if it's just water addition or water plus nutrient additions. And the community increases each time more with every addition. So which organisms are actually allowing this shift? So we looked a little bit, and first of all, we saw that there is a response to water input even when nutrient limitations are not alleviated. So this may mean that water availability allows these dry adapted communities to at least respond to the very low nutrients that are available. We saw that one of the main orders that responded to multiple watering events was the Burkholderealis. So this order is very widespread in cryophilic and oligotrophic environments. So it may be able to function in situ and take advantage of this water and water impulses. We saw an increase in the Actinomycetalis, especially when you're providing them with nutrients. And with the first water addition, there was an increase in chloroacidobacteria. So this group is interesting because it has a potential to do photosynthesis, even if it's not demonstrated in our soils. And we saw pretty much like a decrease in diversity after the third event for both type of treatments. So this indicates probably that only a very minor part of the community would be active when the conditions allow for that, and the rest of the taxonomy that we see in the soils is probably due to just like aeolian deposition, that they are dormant in the soils. A similar pattern is shown for the eukaryotic community. Again, we see that multiple simulated snowfall events and nutrient additions make the community change. And this is especially more evident for just like water additions. And interestingly, we see that freeze-thaw alone has an impact on the community. As we see here, our starting point clusters separately from our freeze-thaw alone treatment. So it may be that some of these organisms are actually responding just to the freeze-thaw alone. Which microorganisms, again, like are responding to this? So the first and most interesting to us is the Cryptococcus. So this is very relevant in our soils, so it's one of the most commonly found. And we saw that it increases dramatically once you give the first water and water plus nutrient addition. So it's possible that it takes advantage like very fast of this water and nutrient impulses. We see an increase in Delphiniomycetes. These are other type of fungi. They may not be as fast of responders, but they are known to be well suited to low nutrient environments. And then the most dramatic shift happened when just water was added. So we get one autotroph, which is like an algae, a Neochlorosarcina, that starts becoming dominant, and another group of fungi, Myceliumycetes, that again are well known for being able like to withstand a lot of stresses. And again, like in the bacteria, we see a significant decrease in alpha diversity with multiple water and nutrient additions. So we saw that Cryptococcus like becomes really dominant as soon as like you provide it with a little bit of water and nutrients. But interestingly, it does so even when it's just exposed to freeze-thaw dry conditions. So it may be that it's partially active when there's an extremely low water content. So we isolated this organism and cultured it and did a phylogenetic analysis, and we saw that it's extremely closely related to another Cryptococcus from the dry valleys, Cryptococcus friedmanni, which is a xerotolerant and endolithic type of yeast. So Cryptococci have been found many times in many glacial and extreme environments. And given its ubiquity in this type of environments and the ability to withstand freeze-thaw cycles, it may be an important functional component of this environment. So what I did next was to try and figure out if the Cryptococcus was not just surviving, but if it was actively growing under these conditions. So what I did is that I set up liquid cultures of it, and I placed them in that same thermal chambers that allow fluctuations. And incredibly, it was able to grow despite freezing solid every night, so with a doubling time of about 2 days. So this, as far as we know, is the first demonstration of growth of an organism during repeated extreme freeze-thaw cycles, and it supports even more our original hypothesis that it has the capacity to grow during this freeze-thaw cycles in the environment, mainly doing so when there is higher soil moisture, which is provided by infrequent snowstorms in this environment. The next question that we want to ask is, which strategies does it use to survive these multiple challenges in this life-limiting environment, and especially how does it cope with freeze-thaw? So our plan is to use a combined strategy of both genomics and transcriptomics to figure out which genes allow survival and metabolic activity in this organism, and to analyze its genome by seeing if it has significant deviations from its mesophilic counterpart. So what we did was culturing it at optimal temperature that we found it to be 17 degrees, and we extracted total DNA, sequenced it, and started its genome assembly and annotation. I'm sorry, did anything happen? Okay, I'm nearly done, sorry. What's gonna happen?
I don't know.
Okay. Yeah, so very briefly, like the genes that we're gonna look for are metabolic genes. So since we suspect this to be like a versatile opportunitor that is able to take advantage of whatever the atmosphere brings it on this high elevations, we expect it to have many degrading pathways. We will look at genes associated with oxidative and osmotic stress. We still don't know what kind of like stress signal is generated with freeze-thaw cycles. Cold adaptation genes necessary to complete meiosis to see if it's able to do sexual reproduction. And then genes that allow to withstand fast temperature switches, for example, related to membrane fluidity, and genes that allow it to live at a low water activity. So we have to keep in mind that when there's low, in this freezing environment, there is extremely low water activity. So they are like very similar to hypersaline environments. And the last step we'll do to do a transcriptomic analysis of specific time points within that thermal cycle. So the idea is to grow it and sample at 8 different temperatures within the daily thermal cycle, do a total RNA extraction, and get its transcriptome sequence to find out which genes are up and down regulated under the freeze stress compared to optimal temperature, and to see if it's active during the whole freeze-thaw cycle. So all this work is relevant to the NASA astrobiology roadmap because it aims to determine the physiological state of microbial cells that are found in extremely dry and temperature fluctuating environments to get more insight on the dynamics of survival of microorganisms, possibly on the surface of Mars. And our work is the first to explore adaptations to external thermal fluctuation by analyzing the genome and transcriptome of what at this point can be called a polyextremophilic yeast. So given its unique ability to withstand a lot of stressors, it is a potential model organism of astrobiology and for stress-resistant studies on eukaryotes. So I want to thank the National Science Foundations and the U.S. Air Force, which gave us the grants to be able to do this work, and the Alpine and Microbial Observatory, which is my lab, that has now become Arctic and Antarctic as well. So if you're interested in looking at pretty pictures of all our sites and our work, this is our website. Thank you.
Thank you very much, Lara. We have time for perhaps one question.
Hi, so I noticed in one of your slides you had something that you called plant material going down when you added water and nutrients, and I was wondering if you could explain that a little bit.
Sure. So I would have to go back. So when we look at this, for example, like we see that our untreated conditions, so just like soils from these elevations have a lot of plant material that we know it's not growing there, but it's blown in. So what we see is that there's a reduction of that plant material. So we have two hypotheses about how that's happening. So one is that Cryptococcus is actually taking advantage of that and degrading that plant material that is blown in, We also saw that it has like a lot of genes that are able to degrade some plant compounds, some plant-derived compounds. Or the other option is that it will simply be degraded as a result of the freeze-thaw cycle itself, because it's known to damage a lot of cells. So it may be eaten up by the Cryptococcus, or it may be just like degraded. But in each case, we suspect that it will, make advantage of whatever nutrient comes out of them.
Okay, thank you very much, Lara.
Thank you.